Okay, so you've definitely heard of serotonin. But what if I told you its real story has almost nothing to do with happiness and everything to do with construction? We're going to peel back the layers of pop science and look at what it really is, the brain's master architect. And trust me, it's a story that's way older and weirder than you think.
So first things first, let's just get this out of the way. We've all been told serotonin is the happy chemical. You know what? That's kind of like calling an entire symphony orchestra just a violin. It's not wrong, but it misses the entire point. Its real job is so much bigger and honestly a whole lot cooler.
You see, the story of serotonin doesn't start in our brains. It doesn't even start with animals. Nope, it starts with the most basic, fundamental engine of life on this planet, the sun. Serotonin's roots go all the way back to the very beginning of life itself.
It all starts with this little guy, an amino acid called tryptophan. Now, tryptophan has this really neat trick. It absorbs light. Billions of years ago, the earliest forms of life were using molecules like this for photosynthesis. But it was also doing something else. It was acting as what's called a morphogenic chemical, which is just a fancy way of saying it changed the shape of things, helping early plants physically turn towards the sun. And that ancient job, changing shape, is the huge clue to what it's doing in our brains today.
So, if serotonin isn't just about making us happy, what is its deal? Well, get this. Serotonin is a messenger that can deliver two completely opposite instructions. The whole secret isn't in the chemical itself, but in who or what is listening. Think of it like this. Serotonin is a letter. The words written in that letter are always exactly the same, but the meaning of that letter, the action it triggers, totally depends on who opens it and reads it. And in our brain, those readers are called receptors.
And a receptor is really just a specialized protein that sits on the surface of a cell. It's like a perfectly shaped keyhole. When a molecule like serotonin, the key, plugs in, the receptor turns and sends a very specific command into the cell, telling it what to do next. And for our story, we really only need to meet two of these receptors. On one hand, you've got the 5-HT1A receptor. Its message is basically, settle down, grow up, be stable. Then you've got its polar opposite, the 5-HT2A receptor. Its message is change, grow, rewire. This is the one linked to psychedelic experiences. It's the same messenger, but two completely different, even opposite outcomes.
So how does that work? Well, on a chemical level, it's pretty straightforward. When serotonin hits the 1A receptor, it reduces an energy molecule inside the cell, basically telling it to power down and chill out. But when it hits the 2A receptor, it floods the cell with calcium, which is like slamming the accelerator. It ramps up energy and activity like crazy.
All right, let's properly meet our first character in this drama, the 5-HT1A receptor. You could think of this one as the brain's force for order, for structure, for stability. Its whole job is to build a reliable, finished product.
So when serotonin binds to 5-HT1A, the message is crystal clear. All right, everyone, tools down. It tells young, dividing cells to stop making copies of themselves and to mature into their final form. It reinforces the cell's internal scaffolding, its cytoskeleton, and just generally promotes a calm, stable state. It's the construction foreman of the brain, shouting, let's get this finished. And you can see that right here. The 5-HT1A pathway takes a young, kind of generic cell and pushes it toward maturation, helping it become a complex, specialized neuron. It's basically creating the final finished pieces of the puzzle. And hey, you don't have to take my word for it. The source research we're looking at sums it up perfectly. The key phrases to notice are intracellular stability, cell differentiation, and cessation of proliferation. It's all about finishing the job and making things solid.
And this isn't just abstract biology, okay? This is exactly how many anti-anxiety drugs actually work. They are designed to specifically target and activate this 5-HT1A receptor, basically tapping into the brain's own built-in system for calming things down.
But right, a brain can't just be stable. It can't be set in stone. It has to be able to change, to learn, to adapt. And for that, we need to meet our second character, the 5-HT2A receptor. This is the agent of chaos, the force for plasticity, growth, and total rewiring. So if 1A is the foreman, 5-HT2A is the architect who just showed up with brand new blueprints and ordered a massive renovation. It does the exact opposite of its counterpart. It tells cells, keep dividing, we need more building materials. It actually destabilizes the cell's skeleton so it can be reshaped to form new connections, a process called synaptogenesis. And the contrast here is just night and day. While the 1A receptor was pushing cells towards maturation, this 2A pathway is pushing them towards mitosis, which is just cell division. This isn't about finishing the building, it's about adding a whole new wing.
And again, the research backs this up. The key words here are destabilize, proliferation, and synaptogenesis. But notice that last one, apoptosis. That's programmed cell death. So this change agent is so powerful, it can not only order new construction, it can also call in the demolition crew.
And this brings us to the other side of that clinical coin. The drugs that are famous for powerfully activating this receptor are the classic psychedelics. By massively cranking up plasticity and disrupting the brain's stable everyday patterns, they trigger those profound shifts in perception that we call a hallucinogenic experience.
Okay, so we have a stabilizer and a change agent, a foreman and an architect, a calm down signal and a shake things up signal. How do these two totally opposite forces work together without tearing the place apart? Well, this is where the real magic happens. It is a beautiful, delicate balancing act.
And this really shows you the two forces at work. On one side, you've got 5-HT1A taking these dividing cells and turning them into mature, specialized brain cells. On the other side, you've got 5-HT2A telling those same cells, nope, make more copies of yourself, fuel more growth. It's this constant fundamental push and pull.
And when you lay it all out, the opposition is just so elegant. You've got differentiation versus proliferation, stabilizing the structure versus destabilizing it. One receptor even tells support cells to release chemicals that promote maturity, while the other tells them to break down sugar for raw energy. It's a perfect yin and yang, playing out at every single level of the cell. And it all comes together in this beautiful, dynamic equilibrium. Serotonin, working through these two very different receptors, is orchestrating a constant tug-of-war on the physical skeleton of our brain cells. And this balance is what allows our brains to be both stable enough to have a personality and plastic enough to learn something new tomorrow.
So if you take one thing away from all this, let it be this. A healthy brain is never finished. It is not a static object. It's a living process. It's a continuous, elegant dance between holding its shape and being willing to reshape itself. And this one ancient molecule is the conductor of the whole orchestra.
And that, of course, leaves us with a pretty fascinating question to chew on. For decades, we thought about mental health in terms of just turning up the volume on a chemical. But what if the real answer is much more subtle? What if the future of neuroscience isn't about boosting chemicals, but about learning how to gently, carefully fine-tune this ancient, delicate balance between stability and change?